My roll number is 61 and I'll be explaining about mutation. What are mutations? Mutations are changes in a genomic sequence, the DNA sequence of a cell's genome or the DNA or RNA sequence of a virus. They can be defined as sudden and spontaneous changes in the cell. Mutations are caused by radiation, viruses, transposons. Transposons means that it is a DNA sequence that can change its position within a genome and alter the cell's genetic identity and genome size. And mutagenic chemicals. Mutagen means that it is a change in the DNA of the cell, and one of the example of mutagenic chemicals is diethyl sulfate, as well as errors that occur during meiosis or DNA replication. They can also be induced by the organism itself by cellular processes. Cellular processes is that it is carried out at the cellular level but not necessarily restricted to a single cell, such as hypermutation. Hypermutation is a mutation that is abnormally frequent. Mutation can result in several different types of change in sequences DNA. This can either have no effect, alter the product of a gene, or prevent the gene from functioning properly or completely.
Type of mutation. The sequence of gene can be altered in number of ways. Gene mutation have varying effect on health depending on where they occur and whether they alter the function of essential proteins. Type of gene mutation. First is point mutation. It is often caused by chemicals or malfunction of DNA replication. Point mutation are of two types. First type is transition. Most common is the transition that exchanges a purine for a purine or a pyrimidine for a pyrimidine. A transition can be caused by nitrous acid or mutagenic base. The other type is transversion. Transversion is less common. In this, exchange of a purine for a pyrimidine or a pyrimidine for a purine is done. Example of transversion is adenine being converted into cytosine.
Hello everyone, my name is Suruchi, roll number 53, and I am going to talk about insertion mutation. Insertion mutation is addition of one or more extra nucleotide into the DNA, which are usually caused by transposable element. A transposable element is a DNA sequence that can change its position within a genome or error during replication of repeating element. Insertion in coding region can alter splicing of the mRNA or cause a shift in the reading frame. Both can alter gene product. Insertion can be reverted by excision of the transposable element. In the given figure, we can see that original DNA sequencing contains specific DNA bases which code for an amino acid sequence. Insertion of single nucleotide, suppose A, in the DNA sequence may lead to incorrect amino acid sequence, and hence there will be production of malfunctioning protein.
Mutations remove one or more nucleotides from the DNA. Like insertions, these mutations can alter the reading frame of the gene. They are generally irreversible, though exactly the same sequence might theoretically be restored by an insertion. Transposable elements able to be revert a very short deletion, say 1 to 2 bases in any location, are either highly unlikely to exist or do not exist at all. Note that a deletion is not the exact opposite of an insertion. The former is quite random, while the latter consists of a specific sequence inserting at locations that are not entirely random or even quite narrowly defined. Deletion mutation. The original DNA code for an amino acid sequence is C80 along with amino acids which are histones. In deletion mutation, a deletion of single nucleotide takes place, an incorrect amino acid sequence which may produce a malfunctioning protein.
Hi everyone, my name is Margaret, roll number 16, and I'm going to talk about frame shift mutation. A frame shift mutation is a mutation that was caused by insertion or deletion of a number of nucleotides that is not evenly divisible by 3 from a DNA sequence. This insertion or deletion can disrupt the reading frame on the grouping of the codons due to the triplet nature of gene expression by codons. This deletion can be caused by errors in some chromosomal crossover during meiosis that causes several serious genetic diseases. Deletions that do not occur in multiples of 3 bases can cause a frame shift by changing the 3 nucleotide protein reading frame of the genetic sequence. A frame shift mutation occurs when a protein is drastically altered because of an insertion or deletion. Tay Sachs disease is the example of this frame shift mutation. It is a human disorder caused by this mutation. This type of mutation changes the way that codons are read and results in a change in the final amino acid sequence. A change like this can damage the final protein structure and ability to function correctly. Effect of frameshift mutation. They are extremely likely to large-scale changes to polypeptide length and some chemical composition, resulting in a non-functional protein that often disrupts the biochemical process of a cell. They were called frameshift mutation because, as we see from our diagram, the essentially shifted, and this shifting occurs because of the editing of the codon. Lastly, mutation can also be caused by some radiations and that cause DNA to break down. So the cell would end up with DNA slightly different than the original DNA and hence it's called mutation. Thank you.